Welcome everyone. So we will have 5 people and they will have 10 minutes each at the most. Hopefully we will have 10 minutes at the end to discuss a bit. We have from your right, we have Virginie Bros-Feyser. She's representing Eurordis. She's based in Brussels.
No, Paris.
Paris. But you still like Brussels.
Yeah.
Anyway, so, and then next to her we have Ayola Adegnika from ARNTD, Cermel in Gabon. And then we have Paula Rio from CIEMAT in Madrid. And Filipa Ferraz-de-Oliveira from the ERC in Brussels. You are still in Brussels?
Yes.
And then, last but not least, we have Lionel Cavicchioli from The Conversation. He's a journalist here in Paris. So, since I understand Filipa wants to leave earlier, so you could start. And yes, I could put the slides. You can talk. Okay, good. Okay, Filipa represents no, you don't need this. Filipa represents the funding agency, ERC. Good.
Okay, good afternoon. Since I only have 10 minutes, I will start immediately. I was asked to speak about the concrete perspectives for patients, but about how to grant research in this area. And since I'm working in the ERC, European Research Council, that is part of the European Commission bodies, I'm speaking about the way the European Commission funds this kind of research. So the first very, very important message I have to you is that in fact, European Commission funds research in these areas, following the Oviedo Convention article, and that means that we cannot modify the human genome only if it is for a preventive, diagnostic or therapeutic purpose, and we cannot introduce modifications in the genome of the descendants. So following this article in the Commission, we can fund this research. We have another limitation that is linked to this one, is that we cannot destroy embryos, and so the research funded by the Commission cannot destroy human embryos in this area. Apart from that, even with a much longer and complex process to get an authorization, we fund research with human embryonic stem cells and with gene editing also. So, I tried then look a bit to the past 5 years, 6 years, to see where do we have this research being funded, in which programs, because in fact we have a lot of programs in the Commission. I'm working in a small one that is here in the corner, ERC, European Research Council, but we have a lot of projects funded in other programs, and that's why I put here the ones where I found more research in this domain. And apart from the main programs of RTD, the Research and Innovation Direction, where we have research designed or granted in a top-down way, so defining priorities in health, demographic change and well-being, food security, and so we have then the bottom-up programs like ERC and Marie Curie, where we find also a lot of this research funded. We have different types of actions, and for that, my message is that the large majority grants 100% of the costs of the research, and that is also very important to the researchers. And you can see there that we have research and innovation actions that are usually given to consortium, in some cases big consortium. But we have also innovation actions that should be funded with the participation from the applicant. We have coordination and support actions that are designed to support the research line that is being developed. And so those are much smaller grants but are funded in a 100% rate. Then we have the European Research Council grants. It's the agency where I work, and here is 100%. And those are probably the ones that give more money per project, in fact. And there's another special thing is that the usual programs from ERC are for 5 years. And so we do not have immediate results. We will have them a bit later. We have the Marie Curie actions and co-found actions where the participation is not fixed. It's a co-fund. And then we have these SME instruments that are, well, given like lump sums and with a specific aim when distributed. The reason I'm describing the type of actions here is because then I will show you what happened in the last years. So I went to the projects that are already public in the projects granted by the Commission. And here I must say that a lot of them only go to this database when they finish. And that's why there's not a lot of ERC, because they are not finished. The ones that started in 16 are still going on, and so they are not here represented. But I went to these different type of grants, and I checked it since 2015, so 4 years ago, and I found immediately around 100 projects in this domain. What is maybe even more than I expected, because for my experience point of view, we have seen much more in the last 2 or 3 years, also in ERC, and those are not yet here registered because they did not finish. So you can see that there's a lot of Marie Curie projects there. They are much shorter in time than the ERC, and so they appear immediately. But I also looked to see where is the host institution that manage these projects, to see if they are going all to the same place, in fact, this money to do this kind of research. And looking to the coordination, because we have consortia and we have individual researchers in the projects. In fact, we have more projects in Germany, United Kingdom, Netherlands, and then it starts. Spain is still, and France in the top countries, and then we start having, but we have a lot of countries receiving money in this area, what means that we are distributing to all the countries there. The starting here, it's not so visible. Yeah, I got some few projects from 2014, but in fact we had much more projects submitted using CRISPR, and so from 15 on. And so we have 15, 16, and then when we go to 17 and 18, we have already the impact of the projects that are not finished, because the usual dimension in the Commission in general is 3 years, and so they did not finish, they did not register here. And the ones of ERC is 5 years usually, so they do not appear there. This is not an exhaustive list of projects, and that is not the purpose. The message here is only telling you that, in fact, this is going on, the funding of research in this domain. And so then I tried looking to all these projects to see in which specific domain the projects are. And here you can see that we have a lot of things, but we have a lot of fundamental research here that is there trying to develop infrastructure in structural biology. And we have a lot of them concentrating only in diagnostic tools, but not precisely in a concrete disease. But we also have, and we have also the regenerative therapies and so, and chromosomal therapy and cellular therapy, even if it is not concentrated in a disease. But then we have a lot of projects already that are clearly turned to specific diseases, infectious diseases, cancer, diabetes. Then I put together a lot of them that I found only with one study per disease. But we have also a lot of research linked to nutrition and in some cases, even with the production of the of the food that is consumed, and that is linked then to the nutrition and health disorders. Well, some projects in antibiotic resistance and some in aging in general. So this is my main message today, very short, but only to tell you that in fact the European Commission is granting research in these domains, and there's much more there running that does not appear here because it's not public yet, and so I didn't include it. And well, I'm open to questions, and we finish.
So I think I'll be less than 10 minutes, probably 5. But just made a few points. So I think for us first, I mean, when we were being asked what's the concrete perspective for patients, was that the title? Well, first, I think we all need to agree exactly what we're talking about. So, and we've heard throughout the day about the different things, but I think it's so important to really differentiate whether we talk about somatic versus germline. Both actually obviously have, you know, benefit and concrete implication for patient, and also research versus clinic. Again, both have very strong implication for patients. And I just wanted to go back a bit maybe to because we talked a lot about germline and potential therapies, but actually even just, and thank you, you mentioned that you are actually funding, you know, project using genome editing to develop diagnostic tools. And I think that's also another important in research, but then, you know, in clinic as well. The whole potential of that technology, not just to develop therapies, which except for ex vivo is really maybe in the future, but the potential and the added value that we have now, really, using genome editing in research to develop diagnostic tools, but also, especially for rare disease, what's really important for us and for rare and undiagnosed diseases, when you have very rare diseases for which, you know, even exome sequencing didn't really give you an answer for your disease, then you have maybe identify a few mutations, the VUS, the variant of unknown significance, but it's difficult to really just, what do you do with that? I think the genome editing tools also offer hope in that you can do functional studies quicker maybe, as well as developing disease models. We heard a bit about the mice, and I think also from the gentleman earlier that working in zebrafish. But I think all of that is also really important to convey to patients. So obviously education and information to make sure we understand and we are talking about the same things first. But regardless of that, in any case, we have, as long as we have an unmet medical and health needs, we have an imperative to keep exploring. And that means that when we have tools potential that have, you know, potentially really efficient and accurate, even though that's a blurry area still, but then we have the duty to really ensure that they are used to the best of their potential for the benefit of patients, to advance research, and obviously to understand the pathogenic mechanisms and develop ultimately treatment and therapies. So another key point, I think, from the perspective of patients is to manage expectations and so to inform patients on the progress regarding the safety and the efficacy of the tools. And I mentioned earlier in the breakout this morning as well that, you know, considering all the hype and that 25 years ago, especially, for example, if we take Duchenne patients, they were being told that within a couple of years, gene therapy was going to be it for them. They're going to have curative treatment, and then here we are now, and we're still not quite there. It's actually a different stories. So really often patients ask us, so what's different? You know, why now there's the whole hype? Is it going to be all, the same again? And so again, something else that we talked about this morning during the breakout is actually what is different to what's really truly specifically different to genome editing. When some of this, for example, some of the issues is if we consider the scope of treating or preventing genetic condition, so much is actually similar to somatic when we talk about gene therapy. And also germline, for example, is quite similar to maybe IVF and PGDs. So what's really once we've established that it's important to maybe map out the specifically new issues around genome editing and what's similar, what's not, then we can actually maybe start to be practical and non-hysterical and just go back, turn down the hype and start to be practical and speak and discuss in terms of practical, concrete cases. Put it always back into a context, because otherwise for patients, they won't care. It has to be really in a concrete context, and there's loads that we can actually discuss, especially for rare diseases. So really patient information and education is key to start. We can't ask anyone really to form an opinion or to have a position if you don't tell them what we're actually talking about, and to give them accurate, up-to-date, and as much as possible unbiased information on the current state of play using genome editing, whether it's research or in the clinic. So I think that was my point, yes, but hopefully maybe we can discuss further a few of them and others. Thank you.
Thank you.
So, thank you for the invitations. I have slides. So my idea was to talk about the possibility to use gene editing and the perspective. Sorry, you're not hearing me. I'm a bit small.
Close to the microphone. Close to the patients and
Wait.
close to the microphone.
No, this is okay. Okay.
You can do
No, because I move a lot my hands, so that's okay. Okay, so do you have the pointer? Yeah, thank you. So, and this one, can you hear me? Maybe it's better from this one. So from the perspective of a team that is working, as Luis has told you, in gene therapy for patients, okay? So I think that, as it has been previously mentioned, it's very important to differentiate the type of therapy that we are applying. So I have added this slide in which when you think about gene therapy now, gene therapy with viral vectors and also with other strategies is working for, I would say, for many disorders. And we have to differentiate if we are applying the ex vivo or in vivo strategies to conduct gene therapy. In my opinion, the ex vivo gene therapy is expanding faster because there are no risks to target other tissues, always when we are talking about, of course, somatic cells. And this is especially true in the case of the hematopoietic disorders that are affecting the blood, because there are very good protocols to obtain the cells from the patients, correct them in vitro, and reinfuse in the patients. For example, in the case of the brain or the liver, you have to deliver directly the therapeutic strategy in vivo because you cannot culture the cells in vitro. And this is a reason why the hematopoietic disorders have been, or the gene therapy in hematopoietic disorders is going faster than in other disorders. The strategy in general in this type of disorders is to obtain the hematopoietic stem cells from the patients. You can correct them in vitro and reinfuse in the patients without any problem, because this is something that is usually conducted for the allogenic transplantation in the patients. Why do we select these cells that have this strange name? It's because these hematopoietic stem cells are the cells that can generate all the cells from the peripheral blood. So all the different cell types in the blood can be generated from these cells. So if we can correct these cells using an integrated system, all the cells that are derived from this will be corrected. Okay. So this strategy is very important to say that it's also is only used for the treatment of severe inherited disorders, as the case of the immunodeficiencies, hemoglobinopathies, and also bone marrow failures, and in those patients that don't have any other possibility to be treated. Okay? And as we have also discussed today, we always have to evaluate the risk-benefit to treat these patients. I am going to talk initially about the strategies that we are using nowadays in the lab, and most of the groups are using for correcting patients' cells in clinical trials. That is the use of non-targeted gene therapy. In general, to correct inherited disorders, since we need the correction for all the life of the patient, we need to integrate our gene, the wild-type gene, in the genome. this is done in the hematopoietic system using integrative lentiviral vectors. These vectors can integrate a wild-type copy of the gene in any region of the genome. So we don't really know where it's integrated. Of course, to analyze, we have always to analyze where it's integrated by after we correct the cells, okay? So as is written here, it's not possible to target the transgene to a specific locus, and also we need to include a promoter to express this gene. So this means that the endogenous expression that in some cases can be essential for the expression of the gene, in this case can be lost, okay? Anyway, this seems that everything is terrible, but when we analyze the patients that have been treated with this strategy, we have many disorders, especially immunodeficiencies, in which the efficiency of this strategy is really high. And we haven't detected any side adverse event. Of course, because many preclinical studies were conducted with these lentiviral vectors. So the message is that gene therapy for many disorders is working using integrative lentiviral vectors. But when we had the possibility to use gene editing, I think that we thought this is wonderful, because we can integrate the gene where we want, specifically in a place. We can control where this integration is occurring. This could increase the safety of the therapy, as you can imagine, because in the case of the patients treated by gene therapy with lentiviral vectors, we have to follow the patients for 15 years, analyzing the integration pattern every 3 to 6 months to control that there is no problem. Although, as I didn't mention, but in the previous slide, there are patients now treated for 11 years that didn't develop any problem with the lentiviral vectors, but still we have that concern. And of course, in the case of the specific correction of the mutation, we can maintain the endogenous regulation, something that is very important for some genes. So when we think about using gene editing for correcting disorders, we always think about the use of a donor template and the homologous directed repair. But the efficiency of this system is lower than using the non-homologous enjoining to interrupt genes, as we have talked. So moving to the clinic, this strategy is the one that is going faster because the efficiency is higher and the approach is much simpler. And when we talk about hematopoietic stem cells, even more because the frequency of this strategy is much higher in the hematopoietic stem cells. So I only want to explain you 2 examples that are nowadays in the clinics using gene editing for the beta thalassemia and the sickle cell disease, in which this is a disorder that is affecting the production of the hemoglobin in patients, both disorders, and the number of patients is really huge. The patients have suffer a lot of transfusions and also many different pains, and if they are not treated, they can have an early death. So for these both disorders, a gene editing strategy has been applied to produce. So in the case of the hemoglobin in humans, we usually have a production of the fetal hemoglobin that disappears once that we are born and starts to appear the beta globin. These patients have a defect in the beta globin, so the idea is to maintain the expression of the beta globin in the patients by interrupting a gene that is blocking this expression. Okay? So this is nowadays in clinical trials, and as I previously said, the protocol is very similar to other hematopoietic disorders in which the hematopoietic cells are collected from the patient. They are edited, interrupting this BCL111 sequence, and the cells are reinfused in the patient. Of course, after many analyses to test that these cells are, in theory, and as far as we can know, safe for the patients. So, as I mentioned, there are now 2 clinical trials using CRISPR-Cas system with these strategies, and the only result that is now published is that the first patient that was treated with beta thalassemia now is transfusion independent, while he was previously transfusion dependent 4 months after the treatment. So the initial results are really good, and I would say that even better than with the lentiviral vector, at least with the initial results with the lentiviral vectors. But this will be relevant studies also to test the safety of this strategy in patients. what we are doing, as Joyce was mentioning, is working in another bone marrow, another hematopoietic disorder, that is Fanconi anemia, in which this is a rare disease. The prevalence is quite low, but in Spain we have around 200 patients, and the genes involved in the disease are implicated in DNA repair. So the clinical phenotype of these patients is very, they have congenital abnormalities, cancer predisposition, and also bone marrow failure. And this is the bone marrow failure, the main cause of death in these patients. For this reason, this is the main, this is the first thing that has to be treated in the patients. They can be treated by hematopoietic stem cell allogenic transplantation, but not all the patients can benefit from this treatment. And also the main problem in the case of these patients is that they are very sensitive to the chemotherapy that is used for the transplant. So they have an increased incidence of squamous cell carcinoma after the transplantation. So our idea and the idea for other groups was to use hematopoietic stem cells from the patient, correct them by gene therapy, initially with lentiviral vectors, and now we are also testing in the lab the gene editing strategies, and reinfuse the cells in the patients. And this is what we have done with the lentiviral vectors, harvesting the cells from Fanconi anemia patients in a clinical trial, and these cells were transduced using a vector that carries the FANK gene. Although we have 22 different genes, the FANK is the most frequently mutated in the patients. Around 60% of the patients worldwide carry the mutations in this gene. So we corrected the cells and we reinfused the cells in the patient in the absence of any conditioning. This will be the advantage in comparison of the hematopoietic stem cells transplantation from allogenic donors. When we infuse these cells in the patients, what we observe is, if this is the analysis of the peripheral blood of these patients, that initially we could not detect the corrected cells, but after 4 or 6 months, depending on the patient, we started to detect the corrected cells, and they had a proliferative advantage in vivo. So since they are corrected, they are growing better than the uncorrected ones. This is something that has not been observed in any other disorder. This is an advantage of these Fanconi anemia cells. Since you have a marked defect in hematopoietic stem cells, the corrected one have a marked proliferative advantage. And this prompted us to test the possibility to use gene editing for this disorder. Again, instead of thinking about using homologous directed repair, we thought about using non-homologous engineering to repair the cells from the patients. But in this case, we don't want to interrupt a gene. We want to recover the function. But why did we select this strategy? Because, again, this is the preferential mechanism in hematopoietic stem cells, and it's enhanced in Fanconi anemia cells because, as I told you, the homologous directed repair is affected in these patients. And also because it's much simpler also for the delivery into the patient, in the patient cells, because we will not need per-stimulation. So in 2 hours, we can edit the cells and infuse again in the patient, and only the nuclease will be necessary. But again, how can we generate mutations here and correct a gene? That doesn't sound very good. This is happening in the patients, in some patients, so the idea was to mimic what is happening in some of the patients that are called mosaic patients, in which the patient has a mutation that interrupts the gene, and due to the high chromosomal instability, sometimes occurs a second or even a third mutation. This means that they can generate a new protein with some amino acids modified, but still functional. So our idea was to do something similar with the CRISPR-Cas system. Yes, so using the guide, if this is a mutation in a patient, using the guide, the CRISPR-Cas system, we generate a double-strand break close to the mutation, and the non-homologous engineering will repair this mutation, and we can generate uncorrected cells that have interrupted the open reading frame, cells that are corrected because they recover the function with an in-frame deletion, and unedited cells. Since these cells will have a proliferative advantage, finally we will have all the cells corrected. And this is what we tested in the patient samples. In green, you have the potentially therapeutic index due to a deletion that eliminates the mutation and recovers the open reading frame. And what we obtained was a marked proliferative advantage in the cells from the patients. And of course, we also analyzed the off-targets, but we have analyzed 20 off-targets. I have here some questions also for the discussion, how many off-targets we should study, and also which is the best strategy really to focus on the off-target activity of gene editing, probably the whole exome sequencing, although translocation should be also analyzed. And that's all. I would only like to thank my team working in this project, the leader of the lab, Juan Antonio Guerre, and all the people from the lab, and of course the patients and families and clinicians that are in the foundation of Fanconi anemia in Spain, and all of you for your attention.
Thank you.
Good afternoon, everybody. So I've been invited to give a patient perspective for gene editing, but I'm not doing this currently, but I will share with you the experience of activity which I'm doing, which is related to an innovative tool in Africa to treat or to control diseases. So in Africa, we have a number of diseases which are afflicting the community. As you can see here, the number of infectious diseases in Africa is high, more than in Europe, for instance. And in addition to this pathologies, we still have also some pathologies like non-communicable diseases, which are also occur in Africa. And we need, of course, the therapeutic agent or vaccine for prevention and so on. But we don't have any technology. This is our major issue. We don't have medicine, we don't have vaccine, we don't have anything to treat them unless we have a traditional medicine which is not precise as we want. Of course, in this case, everything which is very good is very welcome, and we are eager to accept what can help us to control, treat, vaccinate, and treat our patients. So, as you can see here, I take example of malaria among all of these diseases, which is the more deadly parasite in Africa. So this has been presented very well by Nolan, which is very good. So as you can see here, this is from WHO report 2018. You can see that nearly all African continent is endemic for malaria, including some part of Southeast South America and Southeast of Asia. But what's going on with malaria? So the correct burden of malaria is that there's no regression, there's no reduction of malaria prevalence or density, density up to more than 2,000, more than 5 years now. So there is a stagnation of malaria prevalence in Africa, and why this happened? There's a lot of action. Of course, it has been shown that malaria control, vector control, have considerably reducing malaria prevalence and incidence in Africa. But what we are seeing in the field is that this happened. Of course, from 2000, let's say 2 to 2010, we have a reduction of malaria, drastic reduction of malaria, and we all say, yes, we got it. But this was concomitant with a spread of network in bednet, implanted bednet, which is also associated with a vector control. We have also the distribution of artemisinin derivative drug, which also contribute to reduce this incidence. But from 2005 on, we have a re-emergence of malaria. That means we fail somewhere. We don't know where we fail, but we need to have answer to that. That is something which is very important. Today, in this report given by WHO, the number of the people dying from malaria did not change since 5 years now. It's the same number they give us. The difference is nearly not significant. That means there's something which is there. We don't have the action. The all effort against malaria does not work as we would like it to be. So, to control malaria or eliminate malaria and eradicate malaria, there's no magic. We need to make, there's a prevention. And the previous speaker, they talk about it very clearly. You need a insecticide net, a vector control, vaccine, chemo prevention, and environmental improvement. But the last point is the most important. But how to improve environment? We need money. We need it more than just talking, we want to improve our environment. Africa is full of forests, of water everywhere. But where can you get money? And if you do that, we will eradicate malaria tomorrow morning. That's what you did in Europe by improving your environment, which we know the solution. If we clean our environment, we don't need drug anymore, we don't need vaccine, we don't need anything else, because we will chase the mosquito, we don't have mosquito anymore. So for treatment also, we need proper diagnostic. That means we need a good diagnostic, which can allow us to don't miss a diagnostic, the real infection. Today, the rapid diagnostic, which has been shown to be a tool to prevent malaria, have the problem with the deletion of HPV2, HPV3, and today you cannot even trust the rapid diagnostic be available in Africa. We need effective drug. Only drug which is real effective today is artemisinin derivative drug. That means there's need to develop a new agent against malaria. If we have resistance against artemisinin derivative tomorrow morning, Africa will disappear from the world. This is the truth, because there is no any drug effective today against malaria apart from artemisinin derivative. And so, and as you know, there is no vaccine. The only vaccine that has been developed is RTSS, which has only 40% of efficacy if we want to be very generous with this vaccine. And currently, it's under evaluation for implementation in 3 countries in Africa. Then before we can deploy that in the continent, we still have to wait up to 5 years or 2-3 years. We don't know what will happen. So the challenge we have is the resistance. Every day, this is a problem. We cannot avoid that. Insecticide and the drugs and so on. I'm happy Nolan showed that with gene driving of mosquito, there's less chance to have resistance. There's no licensed vaccine, as I have said, no adequate health system, and no resources. But coming to put it together, if there's anything like using a CRISPR or gene driving to modify mosquito, to produce vaccine, to produce new drug, Africa will take it. We will take this because we need the solution. We cannot wait and cross our finger. And we don't do anything. We don't have a laboratory. Our scientists are in Europe and we are working together. We are working hand to hand. But the end user of all of this development is not scientists per se, but it's the population. So that's what I say, that if you can use this for this development, for this to understand pathogenesis. So for malaria, vaccine and drug manufacturers, this needs to follow the life cycle of malaria. This is something already we know. We can use this life cycle to try to target appropriate drug and the vaccine against malaria. So what is the requirement for introduction of innovative in Africa? This is not simple. And this is something we are facing every day, because that's what I'm doing every day, try to evaluate vaccine, new drug in the community. You have to face people. We have also to discuss with the stakeholder. The problem starts from the stakeholder, not before you go to the population. The stakeholder want to know, okay, what are you talking about? The safety is the primary endpoint for them. They want to have a safe product, a safe tool to introduce in the community. We need the community to be engaged. The engagement of the community is important, because if they don't understand what you introduce to the community, there is no adhesion. And yesterday, because we have a congress in Africa, in Gabon, in Congo, there's a vaccine against Ebola introduced by Janssen, and where they want to include 500 people in this trial. But people did not commit to this vaccine. How they will evaluate the efficacy of a vaccine where the community is not adhere? This is already a problem. That means it's very important for community engagement, the reinforcement of human capacity. This means you have to train people to understand, like the community, the health community workers, to be able to translate what you want to do to the community in the local world with a good approach. You have to train people. The ethics need to be reinforced. The ethical committee is also another issue in Africa. Of course, we have ethical committee in Africa which is working very, very well. But we need to update them on all of this new technology. We cannot think that the ethical committee which is able to evaluate malaria drug, malaria vaccine, you bring them to the new innovation, they will say, we don't have competence to that. And Eve know that because we worked together, we were together in Congo, where ethical committee from Gabon, from Chad, and Congo, they were there. They don't have any clue of what is going on. And they say, we don't know. We are doing currently the control human infection model is a problem for them. They don't know what that means to which end. We have a regulation problem. The more advanced regulation in Africa is ARRIGE. They have no clue of what is going on. This is also another problem. How do we want to work in the continent with innovation where people who have to give you authorization, who have to comply, doesn't know what is going on? This is a point I think we need to talk about. And the national and the regional commitment is very important. If you talk about the gene driving, as Lionel showed nicely, if you do that in Burkina Faso, mosquito doesn't need visa or passport to cross the border. This is a problem. You have to commit with everyone, including those who are very far away from Burkina Faso, because the mosquito will cross the border. How do you want to contain all of this? This is a problem. Maybe we can discuss that. At the patient perspective, the patient wants to be safe. They will ask you the question, when you in the consentment process, you have to convince the patient that, not convince, explain to him the positive and the negative aspect of what you end for. But you don't tell the patient, okay, this drug I will give it to you, make sure I can tell you will die for more than 60% of chance you will die. Nobody will participate in that. Because this is, we need the safety, innocuity of what to do. The rationale is very important to understand the rationale. Why do you want to do this? It's very clear. The rationale is that we want to eliminate malaria. The rationale is that we want to eradicate malaria, but we have to make it clear for people. Proof of concept to show that it's happened somewhere else. Here is the data. Safety, there's no problem. We can introduce it in your community. You may have the same result as we prove in US or in Europe. We need data, the surveillance system, because when you start, you introduce innovation. You cannot introduce that and run away. You have to follow them up in time to see what will happen later on. All of these points are the perspective which we have to cover anytime we want to introduce the innovation in the population. I know many laboratories in Africa working on Ebola, Zika, Lassa fever to try to develop a vaccine and diagnostic tool. All of this will go through all of these requirements at some stage. Believe me, that is what we are doing every day in Africa, because as researcher and also stakeholder, you have to discuss with ministry every day, ethical committees and the regulatory authority to explain to them the need, but they don't make effort. We have to tell them. It's our business. Thank you very much for your attention. I'm ready to answer your question.
Good afternoon. So, as the previous speaker noticed, the adhesion of patients and the engagement of community is very important when it comes to. You don't hear me?
Louder.
Louder? Okay. I'll take this one maybe.
No, it's a matter of. Hello.
Sorry. So, as the previous speaker noticed, the adhesion of patients and the engagement of community are very important when it comes to treatment and to the acceptance of new research. And in this era of fake news and of defiance against the media, it's very hard to achieve this. And so it's very important for people to be convinced that the information that they get is an information that is solid and verified. And we are experimenting a new way of disseminating knowledge at the conversation for something like 7-8 years now, and 4 years in France. We are working closely with the people that are producing science, you all here in this room, and we are trying to bring some new way of interpreting the news through the prism of science. So how do we do that? We are a collaborative media, and we are associating academics people to senior journalists in order to produce texts that are aiming to decrypt actuality with something rational as possible. So I'm sorry, I was a bit.
You want to go back?
Yeah, no, it's okay, it's okay. And the main idea is that people are doubting that what they hear is something true when it comes from the traditional media. And so by being transparent on who is speaking on the conversation, we are hoping that they will regain confidence in the news. And we are independent, we are non-profit, so we can be completely transparent to our audience, and we are completely free from any pressure because we don't have any ads to sell or any we don't have any stakeholders to satisfy. So we aim at enriching the information landscape and maximize the impact of research by giving more space to the academics. It's something that we are doing now for the last 4 years, and surprisingly, the results are very good, even if we consider the point of view of our other media. I mean, the reception of the conversation was very good, and even the other people that are working in the media field are taking our information in their media. I mean, we were not sure that it would be so because the press is not in a very good shape, as you all know. But it's something that is occurring. And what is very interesting with the conversation is that we really want to show to the people how the science is being constructed. I mean, it's not only about being very. It's not about selling magic. It's about selling a way of thinking, a way of rationalizing the world, a way of looking at the information we are submerged with, and being able to have a critic eye on it. So this is what we are trying to do, and I think on topics just like as complicated as CRISPR-Cas9 or vaccination, for example, or it's something that is very important to do. That's it for me. Yeah.
Okay, thank you. So thank you all. I think we managed to be on time, and even though we started 10 minutes past, so actually we have the 10 remaining minutes for a bit of a discussion. So we heard that this research can be funded, at least in Europe, and we heard the demands from the patients' organization. They want to handle the expectatives. They want to understand how different is this technology from others. We have heard an example of a therapy. We have heard that things that might be working in Europe, they're different in Africa. And now we heard that there are some channels, we have open channels in which we can convey this information. Yes, please. Question, Jennifer. Please.
This is a question for the conversation. I see you're going to you're going to start publishing or being online in Spain. Do you have other projects, other countries, and are you able to follow your readership and see the volume of readership, and what is the interactivity that you have with your readers and that kind of stuff?
Yeah, thank you for the question. Actually, we are a network of newsrooms, and the network started in Australia in 2011, and several other newsrooms opened in the next years, and so now there is a newsroom in Australia, UK, USA, France, Indonesia, Spain, Canada, and South Africa and West Africa too. It just opened last year in order to cover specific issues and topics in this part of the world. And so we work together every day. So every day each newsroom publish the list of the topics that will be published on our websites, and then the other newsroom look at it and say, okay, this one I will take it and I will translate it to Indonesian, to English too. And so that's the way we work. And concerning our audience, it's growing since the last 4 years for France, because we opened in 2015, and it's still growing. And what is very interesting is that we have a newsletter, and the newsletter is still growing, and it's read a lot. It means it's sent, and people don't only put it away; they read it, and it's still growing. And so we are very happy, because it's our papers are quite sometimes it's popularized. We want to popularize science, but we don't want to sell it at a low cost. So you have to put a little of effort in when you read our paper, but and people are happy to do that. So.
Actually, if I may, I'm often writing for Conversation Spain, and these topics are very well read and very well disseminated. So you're doing a wonderful job as a
Thank
channel.
you very much.
So, and they are looking, and every time there is a topic that is worth discussing, they're trying to get the researcher that is closer, and they get involved. And I believe this is happening in all different countries.
I have a question. I think the microphone just for Filipa, because I know that you are in a rush. The results you have presented are the projects funded, selected. That's right? Or all of them?
Yes, all that projects are funded.
Okay.
And we have a lot more funded that are not yet there.
That's the point of my question.
Yes.
You remember in March 2018, I was asking whether sometimes there are going to be some research on science and human and science, sorry, it's very late now, social humanity on Gene Drive. Have you seen some of them, even though they were not funded?
I cannot tell you like that, because I've seen so many things. That
Yeah, but in this field, I mean in this field,
yes.
This field is not. I think it should be very rare, but
what I can tell you is that I used some criteria to search for the projects, and I used the criteria of human genome editing in the last years, and I only picked the projects that are already public. And so, there's a lot of things going on that are not yet public.
It was not a trap, it was just...
No.
Also, I'd like to combine these comments from Steve early on this afternoon, when you said that there's a bunch of clinical trials coming on that we're still not aware. So here we had some examples coming from Spain that they're already showing us that even though we need to remain careful and we have to be step by step, genome editing is already impacting in the clinic. So probably more than what we know, and soon it's going to be much more disseminated. So please go on.
So two things. First, a comment on the conversation. It's true that the conversation in France has been specifically engaged in making CRISPR, in trying to make CRISPR a public issue. Aline Richard and Benoît Tonson have been working in that direction. They've asked for articles for me, for others as well. There's that, and Nathaniel was just there. So Le Monde as well has really tried to make that very much a public issue. It didn't really happen, but it's not the media's fault. And that's what I'm saying back to educating the media. I think in France and probably in the US as well, I can't say for other countries, but the media is very well educated in this sense. Like, they've really tried to get into what CRISPR is, what genome editing is, and they really took the time and effort to make that happen. And probably the problem in public engagement is not in the media, but where what people do with the articles and the information that is produced. I have a question for you, sir, about you've made a presentation talking about what happens in Africa, but it's a very big continent, and like you have very different societies and very different issues in those societies. Probably Northern Africa, countries like Morocco, Algeria, Tunisia will be more interested in sickle cell disease than in malaria. Probably other countries have different problems, such as having sub-regions whose territory they cannot completely control, so maybe they want to try technical solutions on certain territories, and other territories they don't need so much. So in your expertise, in your point of view, what would be the precise surgical approach what African country and sub-region do you think is the most likely to have a enlightened, publicly supported introduction of genome editing technologies? Where do you think this can happen in the best way and with the most community support? Where do you think people want to have this implemented for their own good? In what precise country, in what precise population?
Thank you for your question. First of all, what I talk about, I just pick up malaria as pathology, as diseases. I could choose Lassa, I could choose something else. And the problem I raise is a problem of ignorance of population. So you talk about media, the education of the media, who inform very well. That is the same process. If people does not understand what you are doing, you do it for nothing. Because if in Gabon people are well educated, I can tell you, you don't need to convince them. You just need to explain to them what you want to do, and they can tell you yes, I agree or not. But yeah, there's a community in other country where the people are less educated. How do you want to conduct a study in this area? Imagine you want to translate informed consent into the local language. You're looking for the right word, and your sponsor doesn't not understand your language. You wrote whatever you want there. And I'm laugh after the sponsor who say, okay, or the ethical regulations say you have to write it in the local language. Okay, the sponsor does not understand what it means. In Gabon, we have this chance, everything can be written in French. Unfortunately, we have to use French for our activity. This make it even the best country where this can happen. What I present is that the process is that to explain to people, starting from stakeholder. If you want to introduce a project in the country, you have to discuss with an ethical committee member. It takes time. You have to convey them to the workshop, explain to them what they think. Feedback from them is very important, and you go back, take this in consideration. The social scientists will tell you what that means. I'm not a social scientist, but it's a process to get them engaged. Now you go to the regulatory authority, and in Africa we believe when the WHO tell you it's okay. We don't have, it's a pity that people want to listen to what WHO said. If WHO said it's okay, most of the government in Africa, Sub-Saharan African, will go ahead. But if WHO say no, even it's good, they will not go ahead. This is another commitment that I'm saying, regional and national commitment. If you go to regional, in Central Africa region, you go to OSIAC, which is our regional body for health. And then you go to a WHO, there's ARRIGE. All of these organisms should give their word. Just go to them, to WHO. If WHO agree to go ahead with case by case with gene editing driving, you will go easily to Africa. We need it. We need this technology. Believe me, we need it. But if we don't prepare the field, despite the technology is there, we cannot assess the community. We cannot reach our target. That is the problem. Everywhere you can do it, but we need to explain to people communication. As the journalist said, it's important for people to understand. You don't need to hide. Be open. Tell them the risk and the benefit of what they do.
Thank you. I have a question for you, Virginie. You said you want to understand how different is genome editing from gene therapy that was already acting. So are you saying that we as a group, as ARRIGE, we need to make more efforts to make these things simple, to just communicate? I mean, what is the point? We're missing information. We are not, because you probably don't want to hear from us more data. You probably, what is exactly needed from your perspective?
So sorry, I think I was you misunderstood my point. It's not about giving us more information. It's just what we hear, for example, whether it's in the media or even in workshops sometimes where you're invited and they want to talk about the ethical issues around genome editing. And actually what they talk about are not new issues. We've talked about that when gene therapy first came, so it's... The point is I think that with the different progress and the promises around genome editing, some of those issues which are not new, most of them, are coming back to the surface, are a bit more prominent and on the forefront of the discussion, but they are not inherently specific to genome editing. So just if we strip that away, then what's left, and how can we be practical and just move on to what's new and, you know, not start again the discussion that we had 10, 20 years ago?
Yeah, perhaps, Paula, you can mention what is your experience with the Association for Fanconi Anemia when you explained these genome editing experiments, whether this was well received, whether they understood, whether they got the point, whether they were excited, and I suppose they were.
So I think that the most important is that also depends on the disease. So if you have Fanconi anemia is a disease in which the patients, if they are not treated, they can die with 9 years old. So the parents want to do something to treat the patients. So since we started to collaborate with them, we started the project 20 years ago for the gene therapy with lentiviral vectors, they trust us. But if you read the informed consent or whatever, you think this will never work, because we are saying the possibility to cure is nothing. This is only a trial. You have to be sure that this will not work. You are obliged to do that. Yes. I mean, and we cannot explain something different. And the clinician is the one talking with them, and he tells them, this is probably not working, but you will help someone else with the results. So I think that's. And regarding the gene editing, I think that they are not so, I agree that they are not so educated about the gene editing. So sometimes I think it's a bit complicated for them to learn. But this also happened with the lentiviral vectors at the beginning. So now they see the results with the lentiviral vectors and they say, okay, if you think that this could be better, perfect. But we still don't know if it will be better. The idea is better, but we still need to have results for clinical trials, because lentiviral vectors are working so good in these disorders that I don't know. I expect this would be better, but we have to wait.
I think that ARRIGE had to be the meeting place, the agora, where all the aspects, the scientific aspect, but also the needs, because what we hear from the patients, what we hear from you, Ayola, from Africa, it's that you are eager of taking any solutions for devastating disorders, and I think it's the same for Fanconi anemia, it's the same for terrible rare disease. And the problem that now we are going to face is going to be the cost, because these treatments, because of the customized, individualized therapy, are going to cost a lot by principle, and also because most likely on Fanconi you will have the same kind of request of the manufacturer as we had for amyotrophic muscular spinal muscular atrophy, which means that there is some existing treatments that cost hundreds of thousands a year, and the same way as for SMA, Novartis began by requesting 5 million and conclude by 2 million. For Fanconi, where you have already a treatment which is of a certain cost, you will face the request that will not be aligned on the real cost of the treatment, but on the solvability of the systems you are going to address. So we need also this kind of place, agora, to discuss between the needs, the science, and the real economic possibilities. And in this matter, the media has also their role to to make evident the various stakes that are and the various issues.
So I want to clarify, because in the case of Fanconi, for these patients there is not a treatment, yes. And also the hematopoietic stem cell transplantation is something that is done, but it's very expensive. And the patients have to be in the clinic at least for 1 month. With this treatment, they live in 2 days. So the price is completely different. Yes, it's going down in comparison with the allogenic transplantation.
If you keep an
yes,
hand
when you are
on
talking
the
about big
treatment.
pharmas, that's another issue, I think. And also this is an advantage of the gene editing, because the cost, it will be lower than the production of vectors. The main bottleneck in the gene therapy is the production of the vectors is very expensive. But I think that with the gene editing, this will be reduced.
Okay. So then I think we have to move on and go to the summaries. I want to thank once again everyone in this panel. Great.